Coming up next on mRNA Day, we have an expert discussion on the history of mRNA vaccines with two really esteemed guests. And they're going to be introduced by my friend and colleague, Juliana Lemure, who is Senior Science Editor with Jen. Hi, Juliana.
Hi, Kevin. Thanks so much. I'm thrilled to be here today with Norbert Pardi and Mohamad-Gabriel Alameh. Norbert is currently an assistant professor at UPenn, where he's been working for the past decade. His research interest is the development of mRNA-based therapeutics with a particular focus on vaccines. He works on the development of a novel therapeutic delivery platform using mRNA in LNPs and has used it to generate highly effective vaccines targeting various pathogens. Mohamad joined the Weizmann Lab as a postdoc in 2018. There, he worked on vaccine projects as well as research to better understand the interaction of LNPs with the immune system. Mohamad's also involved in the development of novel proprietary lipids and adjuvants. Recently, he moved into a new role as the director of the Engineered mRNA and Targeted Nanomedicine Core at UPenn. We'll start our discussion today with a short presentation by Norbert and Mohamad, which will give us an historical perspective on mRNA technology. After that, we'll have a chance to ask our own questions. So please put your questions for them into the Q&A section of the Stage tab on the right-hand side of your screen. I know I'm excited to learn more about what Mohamad and Norbert have been up to recently, to hear their perspective on the role that mRNA will play in the future. With that, Mohamad, Norbert, the floor is yours.
Thank you very much for the kind introduction and the invitation. It's really my great pleasure to attend the annual mRNA Day and give a presentation. Today, I'm going to tell you a little bit about the history of nucleoside-modified messenger RNA vaccines. As you all know, this vaccine platform is used in the very successful Moderna and Pfizer-BioNTech COVID-19 vaccines. So I'm going to highlight the most critical findings and discoveries that led to the birth of this new class of drug. So the very first paper that demonstrated that a man-made in vitro transcribed messenger RNA could work under in vivo conditions, so in mice, was published in 1990. This beautiful science paper demonstrated that intramuscular administration of reporter protein encoding messenger RNAs results in protein production in mice. Despite the critical findings of this paper, most people remain skeptical about the therapeutic potential of messenger RNA, and the field mainly focused on DNA-based and other approaches. What were the issues about RNA as a therapeutic platform? First of all, instability. So compared to DNA, RNA is certainly less stable, and it's degraded very rapidly compared to DNA. The second issue was a very important safety problem. If you deliver unmodified RNA into animals or people, it will induce a serious inflammatory response. This is basically a defense mechanism against RNA viruses. The issue is that our immune system cannot distinguish between an RNA that we deliver for therapeutic purposes and an RNA virus that also comes from the outside, and our immune cells sense many different forms of RNA. The third issue was the low translatability and very inefficient in vivo delivery. RNA is a large negatively charged macromolecule, so it cannot really go through the cellular membrane. So it was very important to develop carrier molecules for safe and efficient in vivo delivery of messenger RNA. So the second problem, the inflammatory response problem, was resolved roughly 15 years ago by the early pioneers of the mRNA vaccine technology, or mRNA technology, I should say, Katalin Karikó and Drew Weissman. They published a groundbreaking paper in Immunity, and again described very well how we can make RNA that is not sensed or not sensed very well by the immune system. So I'm going to tell you about the most critical studies shown in this paper. So most probably the first experiment I'm showing is the most important. So what they did, Katalin and Drew transfected monocyte-derived dendritic cells with different types of RNA, so mammalian RNA and also prokaryotic RNA, and most importantly, shown with purple, an in vitro transcribed unmodified RNA. And they measured TNF-alpha production. So basically, they looked at immune activation induced by the delivery of the different types of RNAs. The reason why they use monocyte-derived dendritic cells, because this cell type is equipped with many different RNA sensors. And basically, the figure speaks for itself. The conclusion was that natural RNAs are not equally potent activators of the immune system. So you can appreciate that, for example, mammalian transfer RNA did not induce TNF-alpha production. Other types of eukaryotic RNAs did. And most importantly, unmodified in vitro transcribed RNA induced a lot of inflammation. And so the question was, why is this happening? What is the difference between different RNAs? All RNAs have the basic building blocks called nucleotides. And basically what Katalin and Drew figured out, that the difference between different RNAs is the different types of modifications you can find in RNA. There are more than 100 different types of modifications, for example, methylation of nucleotides, or the most abundant modification is called pseudouridylation. Pseudouridine is a modified version of uridine. Again, you can find it in ribosomal RNA, transfer RNA, in both prokaryotic and eukaryotic cells. So basically what they believe, that most probably the different types of modifications are responsible for the different immunostimulatory profile of different messenger RNAs. And indeed, when they plotted the percentage of modified nucleosides in different types of RNA against the immunostimulatory profile of those RNAs, it really turned out that the higher the percentage of modified nucleosides you can find in an RNA, the lower the immunostimulatory profile of that particular RNA. So you may remember a minute ago I showed you that mammalian transfer RNA did not induce inflammatory response, and it's well known that almost 25% of the nucleosides in a transfer RNA, in a mammalian transfer RNA, that contains modifications. And another example, an in vitro transcribed unmodified RNA that contains no modified nucleosides induced a high level of inflammation. To directly test this, they repeated their studies in monocyte-derived dendritic cells. They transfected these cells with unmodified RNA and also different versions of modified RNA, and they measured TNF-alpha production and another cytokine called interleukin-12. And indeed, it turned out that unmodified RNA induced a high level of inflammation, and different versions of modified RNAs induced a lower level of inflammation or no inflammation. It really depended what they measured. And so this was a very important proof of concept study. A couple of years later, they published another very important paper, and they showed that the introduction of modified nucleosides in the messenger RNA, in vitro transcribed messenger RNA, not only decreases immune activation, but it also increases protein production from the RNA. In this study, they transfected cell lines, 293 cells, or primary cells, murine dendritic cells, with the Ranilla luciferase encoding mRNA. So they use unmodified or different versions of modified RNA, and then they just measured protein production from these RNAs. And again, it turned out that particularly the pseudouridine-containing RNA, there was a lot of protein production from the pseudouridine-containing RNA compared to the unmodified RNA. So if you go back to this figure for a bit, you can appreciate that even if the RNA is modified, it can induce some immune activation. So Katalin and Drew really wanted to figure out what is responsible for this residual immunogenicity and how we can get rid of it. So what they figured out that, or what they suspected, that most probably there is some double-stranded RNA contamination in the in vitro transcribed messenger RNA preparations, and this is sensed by our immune cells. Our immune cells are equipped in many different RNA sensors in different cellular compartments, and there are more than 10 different double-stranded RNA-specific sensors. I just show some of them, and it's well known that if they bind double-stranded RNA, they can induce inflammation, and they can potentially shut down protein production from messenger RNA. So when they tested this, they made unmodified and modified RNA, and then they performed basically a Western blot experiment using double-stranded RNA-specific antibody called J2 to identify double-stranded RNA in these preps, and it turned out that regardless of the modification, they could always measure a large amount of double-stranded RNA contamination in these preparations. So they developed HPLC purification procedure to get rid of double-stranded RNA and other contaminations from in vitro transcribed RNA preparations. And what you can see in this beautiful northern blot, it turned out that if you use pseudouridine-containing RNA and you HPLC purify it after in vitro transcription, you can your RNA won't induce any type 1 interferon production or pro-inflammatory cytokine production after delivery to dendritic cells. And so we could make a very pure and therapeutic quality messenger RNA. So this was the time 10 years ago when I joined Katalin Zendru's lab at the University of Pennsylvania. Again, we could make very nice RNA. It worked much better in cell transfection studies than plasmid DNA, but we really wanted to use this for in vivo studies. So we really wanted to come up with vaccines, for example. We wanted to immunize mice, and the problem was that we didn't really have a good method or a good delivery molecule that we could use for RNA delivery into animals. We tried to use we used naked RNA that was not encapsulated into any reagents. We tried many different in vitro transfection reagents, but nothing really worked well in mice. We were really struggling for several years, and the breakthrough occurred in 2014 when we started to work with a company called Acuitas Therapeutics. This company developed special lipid nanoparticles for basically small interfering RNA delivery. And we asked them to formulate our messenger RNA into these LNPs, and so we could test it in animals. And that's what we did. We sent them some firefly luciferase encoding RNA. They formulated it into lipid nanoparticles, and then we tested it in vivo imaging studies. So mice were injected with these luciferase mRNA LNPs intravenously, intraperitoneally, intramuscularly, subcutaneously, intradermally, even intratracheally. And then we just measured protein production from luciferase mRNA LNPs. And it turned out that LNPs are excellent mRNA carriers. When we deliver mRNA LNPs systemically, we can generate lots of protein primarily in the liver for a couple of days. But when we deliver it intradermally, intramuscularly, we can measure protein production for a longer duration. And this is basically the ideal kinetics for immunization studies. So we published this data in 2015. And 2 years later, we published another very important paper that demonstrated that the nucleoside-modified messenger RNA lipid nanoparticle platform is a really great vaccine platform. And this was the very first paper that demonstrated this. So we developed a vaccine against Zika virus, and we demonstrated that a single immunization with this mRNA vaccine induced protective immune responses in mice and monkeys. After this, since then, we published more than 30 papers in high-end journals belonging to the Nature, Science, and Cell family of journals, and we demonstrated that many times, and of course others as well, that this platform could be used for to develop very efficient infectious disease vaccines. So as Juliana mentioned, I currently am leading my independent laboratory at the University of Pennsylvania. The major interest of the lab is the development of infectious disease vaccines, with particular focus on the development of a universal influenza vaccine using the nucleoside-modified mRNA technology. So in this project, we take advantage on the enormous flexibility of the mRNA LNP platform. Namely, we want to formulate several different influenza virus antigens into a single vaccine to develop broadly protective immune responses. And we already published some proof of concept studies last year. In this study, we put 4 different flu antigen encoding RNAs into a lipid nanoparticle, mRNA lipid nanoparticle vaccine, injected mice, and induced very strong antibody responses and also T-cell responses. And we demonstrated that a single immunization with this vaccine protected against a broad panel of influenza viruses. This was just an example. I'm heavily involved in other projects as well. So I think I'm going to stop here and let Mohamad talk about vaccine studies in the Weizmann lab. So I'm very grateful for my scientific mentors, Drew Weissman and Katie Carico, and of course I'm thank you very much for your attention, and I'm happy to take questions later. Thank you.
So thank you everyone for the invite. And basically here, I would like to present some of the actual work that I do in the Weizmann lab and at the University of Pennsylvania. I'm just trying to change the laser pointer. So my current activities in the mRNA space. So basically, I operate in 3 different themes, governing themes. One of them is the mRNA manufacturing and process development. And in that theme, and it's basically the activities that now we are doing at the core at Penn, is the development and optimization of automated mRNA production processes and optimization of the in vitro transcription reaction to increase both yield and decrease contaminant in the same reaction. And we are also developing new methods to modify the mRNA and improve actually protein expression. The other major, also, I would say, theme of research that I'm involved in would be the development of mRNA vaccines and the preclinical assessment of efficacy and toxicity. Majorly, I lead with Dr. Joseph Zeckeler at CHOP, a C. difficile vaccine, the development of a C. difficile vaccine. And I also collaborate and work on a broadly pan-coronavirus and a broadly protective and a pan-coronavirus mRNA vaccine in collaboration with Chulalongkorn University, UNC, and Duke. And also, I'm heavily involved in development of mRNA vaccine for the elderly with Dr. Haddad and Kuzler and Cancro from Penn and from other universities such as Drexel. So in that space, basically, we study the immune response to the elderly, and then we try to modify the delivery systems in order to improve that reaction in the elderly. And lastly, I'm also involved in the development of novel ionizable lipid, or LNPs, where with my previous PI and Professor Page and Suman, a very talented postdoc, we developed a library of approximately 50,000 new lipids, and we screened them actually for structure-activity relationship. We assess a lot of these candidates for immune responses, and we do also a lot of work on dissecting actually the adjuvant effect on transcriptomic level, especially using spatial transcriptomic, in order to guide further designs of those lipids. So I'll start by just summarizing a bit of each section, just the first 2 spaces that I mentioned. So here I'll be giving some example on mRNA manufacturing. So what we saw is that we took, when I joined Drew's lab, they had a database of a lot of sequences that they were producing over the years with Norbert and other people in the lab. So I asked someone to basically put all these sequences with the yield, and then we started doing some statistical analysis and looking at correlation between the efficiency and efficacy of the reactions, who is the person is doing it during which time period of the year, and the compositions, for example, and the length, so different parameter. And what we found that we could optimize the reaction condition in order to increase yield, for example, if we are changing processes, just by adjusting actually reaction condition for every mRNA. So we could increase that approximately between let's say 10 to 35% for each mRNA. And at the same time, we could see that there is a decrease during that optimization process, a decrease of the double-strand and RNA content between, for example, I'm giving it an example here, and these here, a substantial decrease. We also started playing around with a lot of T7 enzyme polymerases and then optimizing their buffers in order to increase actually yield. And we were able to bring a thermostable T7 from a yield of approximately 20 microgram per unit up to 60 to 70 microgram per unit, just by adjusting the buffering conditions and the ions and the ionic strength of these conditions. We also now have a very particular interest. We're continuing that work, but we have a very big interest in characterizing these shorter abortive product or prematurely terminated products, and these certain products that are generated after encapsulation with LMPs, and that were mentioned also in the EMA document. We also showed, for example, that increasing the temperature or using high T7 increases with or without our optimized condition might increase the yield, but also it increases those short abortive product or prematurely abortive product. So we basically decided to opt out of this method and then go back to the 37-degree reaction and optimize it further to even increase the yield. In parallel to that, we also did robotization of the production process, where we can actually now do multiple up to 8 4 or 8 96 well plates and produce approximately 4 to 5 gram of RNA every 4 hours. And then we are now working on the major bottleneck, actually, which is the purification, and we are automating it with certain robots. The second basically aspect of the research that I'm doing, I'll just be showing some data from the C. difficile project. So for the C. difficile is a gram-positive bacteria that actually is considered a threat in the United States and elsewhere in the world. It's also a pathogen for both humans and animals. And what we decided to do with Joseph Zackler from CHOP is to use a multi-pronged strategy, where we are developing a vaccine that can at the same time neutralizes the toxins that are produced by this bacteria, like toxin A and toxin B, and then also targets certain proteins or certain epitopes on the surface to decolonize and prevent colonization from the get-go. We always, in our approach, we take this development very seriously, because we don't want to develop immune responses that can also cross-react with our own microbiota. So we actually do a lot of studies on the impact of these vaccination strategies on the microbiome, and in order to promote and maintain a healthy microbiota. I'm just showing here few example of survivals. These toxins, I just want to mention that these toxins are very lethal. They kill animals at nanograms level or doses, and what we can see is that with mRNA individually injected at 1 microgram, whether it's prime or boost, 1 and 5 microgram, they offer a high protection or survival rate, especially when we inject those mice with very high doses of toxin that are approximately 5 times the lethal dose 100. And then it's the same thing for the immunization with the mRNA encoding for the crop domain of the toxin B, and we see that compared to a regular protein adjuvanted vaccine, the survival is extremely high. Next, what we did also, we use a very big or a strain of C. difficile that is very toxic and very lethal, and we can see that the PBS group, once we gavage it with approximately 10,000 spore, they die after 2 days, whether the immunized mice, they kind of recover fully after 7 days, and their survival is approximately 100%. What's interesting in this preliminary experiment that I showed you here is that they are surviving, and the toxin levels in their fecal or in their stool is very high, showing that we do have a lot of toxin, but the immune response is maintaining actually protection and survival. So what we wanted to do is to study the inflammation, and I'm not going to show the data here for the sake of time, but we are showing that the immune response is also able to protect against inflammation. And then we added a third component to that vaccine in order to prevent colonization. And in those trivalent type of vaccine, we have the same type of survival and recovery, and even a faster recovery and a better survival. I wouldn't say a better survival because they survived 100%. But when we look at their stool, and I'm not showing them here, there is a decrease a lot of the burden, of the bacterial burden, and we're now optimizing that work. So I just want to also thank everyone in the Weizmann lab and all the collaborators that we work with, and I work very closely with. And I want to thank everyone who's attending.
Terrific. Thank you so much. I think we have time for we just have a couple minutes left. We have time for maybe one question each. But so, Mohamad, let me stay with you for a second. So we're all have become so well aware of the role that mRNA has had in vaccine development, but can you tell us a little bit about how the technology is making advances in non-vaccine areas?
Yes, so there's a lot of interest now in using that technology actually for protein replacement, and most importantly also to express certain enzymes or proteins that are able to genetically modify or do some genome modification. And there has been also some clinical data with some companies showing very high efficiency at editing a gene in the liver. And I strongly believe that these technologies using mRNA will be very beneficial, especially those that are that requires actually one injection, such as gene editing applications. So, Norbert, do you want to add anything?
Yeah, so I think there are many applications that the platform is really versatile. So Mohamad already mentioned a couple of examples. I would mention maybe one more that I think it's going to be really important in the future, and this is the use of the nucleoside modified mRNA platform for monoclonal antibody therapy. So monoclonal antibodies are really important, and they are extensively used to treat cancer patients or patients with osteoporosis or autoimmune diseases. The problem with monoclonal antibody therapy that it's really expensive. So production of antibodies, proteins, basically, it's very expensive, and we believe that if we could use RNA and then code monoclonal antibodies on RNA, we could significantly decrease the cost of these drugs. So I think that's a very important, potentially very important application of the platform in the future.
Yeah, and I would just like to add on top of what Norby said, if we are able also to do knock-ins of certain monoclonal antibodies, or for example of certain receptors, in order to mount immune responses or have CAR-Ts, for example, that would be even more beneficial if we are able to decrease actually the repeated dosing with LNPs. So.
Okay, terrific, thanks. And in our last 2 minutes, I'm going to ask each of you to look into your crystal balls 10 years down into the future. And I'd love to know what role you think mRNA will be playing in our lives, in our medicine.
Go ahead.
Okay, so I'm a pretty cautious scientist, and I don't think that I can tell you what's going to happen 10 years later. I think we see the near future more clearly. Let's say the next couple of years. So right now, the only field where basically mRNA proved that it's going to be extremely promising and important. This is going to be the infectious disease vaccine field. So the COVID-19 vaccines are really, really great, and both academic labs and companies are working on other infectious disease vaccines. So maybe the next hit will be, or big thing will be flu. All major mRNA companies, and again, many academic labs, including my lab, is very much interested in developing more broadly protective flu vaccines, because we all know that currently used seasonal flu vaccines don't work very well. So Infectious disease vaccines, for sure. I expect to see more approved mRNA vaccines for infectious disease in the coming years. Cancer, people talk about cancer vaccines. Currently, the results are not that, the breakthrough did not happen yet. So there are promising results for mRNA-based cancer vaccines and also for other therapeutics, but I think we still need to wait for the breakthrough. But I'm very optimistic. I don't think that we can solve every single problem, medical problem with mRNA, but we will be able to solve some for sure.
Yes, so I agree with Norby, and I strongly believe that the applicability actually of this versatile technology strongly depend on the delivery systems. So the more we develop ionizable lipids or other delivery systems that are very safe, non-immunogenic. So just as a parenthesis, we published with Norby in an immunity paper it was online a couple of days ago, where we showed the adjuvant effect of the LNP. And as I mentioned, we have a lot of work now, and also with collaboration with Norby, at studying different ionizable lipids and their effect, right, in order to design new non-adjuvant lipids. So it's very important to understand that developing new LNPs or delivery systems that are immune silent, that would actually propel that technology to be broadly applicable to protein replacement. But if I was going to have a choice right now, I would say the next thing would be either CAR-T and genetic engineering, because they normally require fewer injection than other type of therapies. That's a bit in a nutshell, but we both believe that it will be broadly applicable and successful.
Oh, terrific. Okay, well, thank you so much. Thank you both for that excellent discussion.